Thank you for joining and welcome. My name is Allison Onure, and today I will be presenting work from my dissertation where I've been considering a new approach to high desert restoration that considers both climate and traits in 7 different taxa, including 2 forbs. So while we are generally used to thinking about differences between different species, today I'm actually going to cover the variation within individual species. So there are endless examples of this kind of intraspecific variation. For example, populations of the same species can have different flower types based on their pollinators or soil types, what I'm showing in this figure here. And some of this variation is also just due simply to genetic drift, and it might be just random. But a lot of the variation has really been tied directly to the plant's survival and has come about through adaptive evolution to local environmental pressures. So local adaptation is common and well studied in the Great Basin. And 2 years ago now, our lab published an article that looked at local adaptation in Great Basin plants in studies from the last 75 years. And we actually found that most experiments confirmed at least one of these 3 signatures of local adaptation that I've listed here on this slide. And the prevalence of plants adapting to their local environment has driven a huge push to use locally sourced seeds for restoration, since we want to use plants with the right traits and the right genetics. And most of this work is really focused on using seeds from shared climate zones. And while climate really matters, and we know that climate is tied to functional traits based on past work, we have still seen that within plant populations that are coming from similar environments, there can still be a huge difference in the traits that they have. So climate isn't accounting for all of the variation that we're seeing across these populations. So there must be some other biotic factors outside of just climate that are shaping these plants, things like disturbance history or gene flow. Our goal should really not be to only consider climate when we're choosing appropriate plant sources, but we also need to think about sort of the functional traits that these plants have that might be helping them improve in survival in these really extremely competitive restoration settings. And in our study, we wanted to measure the impacts that both traits and climate have on survival in these competitive environments, and then consider these traits across populations of individual taxa. So I'll really dive into each of these questions further as I go along. So to address all of these questions, we collected from 7 different plants across the Western Great Basin, where all 7 of these taxa were growing together in very similar climates. And for this talk, I will really focus in on the results from the forbs and the shrubs, but know that for all of these tests that I'll discuss, we perform the same work with these grasses. So using all of these seeds we collected, we then went on to build these competition experiments in the greenhouse where we planted natives alongside cheatgrass, which is the common invasive here, and then watered them to match outdoor conditions for 6 months. And we also grew them without this invasive cheatgrass to be able to see what the impacts of cheatgrass were having on the plants. And it was really dramatic, as you can see from these 2 photos. So while we couldn't perfectly recreate field conditions in the greenhouse, we could confidently say that these native plants were really suffering when they were planted with cheatgrass. And every single species that we tested suffered a major reduction in biomass, both above and below ground, and in their overall height. So some species were reduced over 80% when planted with cheatgrass, and that's only if they survived. So with all these data collected, we were able to answer our first question, which was asking, were there differences in who survived in the common gardens based on where the seeds were collected? And across the board, here are just the average rates of survival across the experiments. So I should note that survival here means you not only emerged from the ground, but you also then lived to the end of the season to get measured. And you can see that Origeron species, for instance, were the big headache, and others were really cooperative, like rabbit brush, which had an overall 62% average survival rate, which is great. So here, let's look at one example using Elemis. So this graph has seed sources where they came from across the x-axis, And then survival is listed on the Y, and the black line across the middle is average. So you can start to see that the answer was yes, populations were really different in how they survived with cheatgrass. I'm not going to show you all the results for every single taxa, but this was really the same pattern for shrubs, forbs, and the other two grasses, but there'd be different populations that did well. So then second, we wanted to see if we could identify any single locations that were producing exceptional seeds across all of the taxa we tested. And if so, that might indicate, okay, there are spots creating seeds that are ideal for establishing invaded systems in the area, which is a very different way of thinking from what we do now. Currently we really use a mishmash of seeds pulled from multiple locations. There really is growing evidence from community ecology that suggests, we might have more success if we use seeds pulled from a single location that was co-evolved. And these sites might be selecting for high-performing restoration seeds, not only because of their climate, but also their site's history or gene flow. So let's start looking at the results from the shrubs and forbs that were grown in the greenhouse. So you can see, yes, some populations overall were either above or below average. So this graph again is similar, showing relative survival on the Y and then collection sites on the X-axis, but now species are shown in different colors, and you can start to see, okay, wow, some sites like Dahi, DHI overall on the right, all of the species were above average, but then BMC all the way on the left were all below average. So there is some biologically relevant pattern occurring here where entire communities were producing above average populations, even if most of them sort of fell in the middle where some were above and some were below at a site. So using the same wild harvested seeds from these past two questions, we then designed a study to answer the final set of questions, looking at how traits and climate were related to survival. So for this, we established a new greenhouse common garden that extracted shoot and root traits for all taxa populations. So our team collected over 7,000 seedlings at very specific ages, and then scanned them to collect all of these fine scale data that I list in this upper figure. And if you're really curious, we just published this paper in Restoration Ecology that really details out how we found these native plant traits and how other people in their systems could also use similar methods. So we found that seed source locations had, without exception, different traits. So where you came from indicated what kind of traits you would have as a population. So it suggests that there is a genetic basis for all of these traits, since where they were coming from was really impacting how they behaved. So here are just two mini case studies. So first was Stipa thurberriana. This graph is showing the different seed sources plotted against the days to emerge. And you can see that some sites like Bald Mountain Canyon, all the way on the left, favored emerging early, whereas North Eagle Lake, all the way on the right, took its time. And then this other figure is showing the difference between the two sides of the root length spectrum for Connectus to glassii, where Gray's Butte tended to grow significantly smaller, it's on the left here, and shorter roots than those that were coming from Austin Summit, which were much bigger. So now I want to tie in climate and thinking about all of this. We tested for trait by environments associations, which is a signature of local adaptation. So I had to split these analyses out by taxa and species, since there were so many differences across the plants. So here are just the results for Connectus as one example. And you can see that there were several very strong correlations. For instance, the populations coming from milder areas, those with higher precipitation, were associated with greater variability in their days to emerge out of the ground and more variability in their size. Also, if we look at the more stressful sites, so sites with higher VPD, which means that the air is able to hold a lot more water and that plants are losing more water during transpiration, so they have more of a pressure to restrict stomata. That's really stressful. Those sites, along with sites with a really strong decline in actual evapotranspiration, those are sites where there was a really sharp transition from rainy to dry season. So these two sites, high VPD, greater decline in actual evapotranspiration, those are stressful. Cheeto at those sites, Connectus to glassii, was actually having larger roots and was creating bigger seeds when it came from these really stressful sites, which is counterintuitive. There was also less variability in root biomass and greater fine root length from sites with higher temperatures, which was interesting. So each taxa, again, had its own unique set of relationships like this. So I can't really make as many broad generalizations here since they were all very unique. So going one step further, I do want to ask my last question, which was, were these seedling traits or any of the environmental variables also correlated with survival, not just with each other? So here are the results for the shrub and forb taxa, where we're considering which root traits were associated with survival with cheatgrass. So this figure is going to be pulling out the top 4 traits that I'll show across the x-axis, and then the coefficient estimate, sort of like how strong of an effect did that trait have on survival on the y-axis. So interestingly, the traits did not all follow the same patterns, but generally we could say that higher seed weight and fewer days to emerge were associated with survival in the field generally. And the variance around these numbers is fairly large, and the model fits really reflect this. They're fairly low. Although you could argue that that's high for such a messy sort of system that we're considering. So finally, let's consider the environmental variables. So now we're looking at the top environmental variables associated with survival. So the climate, what was the climate like at the places where they reflected from? And these were extremely tight models, but there was a wide range in what seemed to predict survival. So for instance, Origeron was the only genera where heat load and soil water capacity came out as predictive. Also, generally, the populations coming from sites with higher minimum temperatures and greater precipitation seasonality were better at surviving in competition with cheatgrass. So now let's just quickly summarize all these results. First, seed sources had significant differences in survival from one another. Then we found that several locations with higher or lower performing seeds across all of the taxa. And then third, we could say that there was definitely a pattern, a relationship between traits and the environment, which is a pattern of local adaptation, as I've said. And then finally, as we just looked at, there were a few seedling traits and environmental factors that were predictive of survival for both the forbs and shrubs. Generally things like higher minimum temperature, greater precip seasonality, higher seed weights were really the traits most associated with survival. So with that, I want to thank all of you for joining, and then hope you're staying healthy, and thank you to both of my co-authors.